Okay, the next speaker is Dr. Gavin Band, and he's going to speak on the genetics of susceptibility to malaria. Now, when I hear malaria, I get very excited because I did a lot of, in my youth, I did a lot of studies on malaria, the chemotherapy, and subsequently, what causes resistance to one of the main drugs. But I'll stop there, Gavin. So Gavin is a statistical geneticist who obtained his PhD from Warwick. And he's now the head of statistical genomics and a group leader at the Wellcome Center for Human Genetics at the University of Oxford. His interests are in host and pathogen genetics, in particular in the interplay between human and parasite genetics in malaria infections. And he has written several software packages for genetic analysis. I think the first speaker showed one of those, and designed the widely used BGen format. He's also teaching co-lead for the GMS DPhil program. Now, over to you, Gavin.
All right, thank you very much for that introduction, and thanks very much to the organisers for inviting me to speak. So my job is to talk to you about the genetics of susceptibility to malaria. I've got kind of two problems, actually. The first is that the talks this morning have been so fantastic, they're going to be a very hard act to follow. So thank you for that, Garrett and Ambroise. And then the second is that I was given a remit for this talk, which was to talk about classical genetic variants involved in malaria susceptibility, more recent discoveries from genome-wide association analysis, and interactions between human genetics and parasite genetics and vector genetics. In other words, I'm supposed to fit essentially 75 years of work into the next 30 minutes of talk. So, better get started. So, of course, if we're talking about genetics of malaria, we're really talking about three things at once. We've got human genetics, and I'm going to draw, for reasons that will become kind of obvious, I'm going to draw humans as if they were red blood cells over here. We've got mosquito genetics that I'm going to kind of not talk about very much, except perhaps to just mention at the end. And then we've got parasite genetics. And so I'm largely going to be talking about this particular parasite, Plasmodium falciparum. This is the merozoite form of it. That's the form that invades red blood cells, but it's really a shapeshifter. It goes through different forms throughout its life cycle. You should be aware that there are other species of malaria that infect humans. Largely, I'm not going to be talking about these. So that sort of cuts down the problem a bit. Here's my kind of rough scheme of the parasite life cycle: invading red blood cells, expanding red blood cells. That's the bit that causes disease, really, and then transmitting through mosquitoes and going around other cycles. So just to put a bit of background on that, malaria is obviously, the reason for studying this is that malaria is obviously a major global health problem. It still is now, over 600,000 deaths due to malaria in 2021. Things have been improving, so here's a figure from the WHO World Malaria Report. Shows you that these green countries are countries that have been certified malaria-free in the last few years. So there's a trend towards eradication happening, and that's very positive, and there are other positive things. For example, a vaccine has recently been approved. There are also very worrying signs. So the plots that I'm showing you down here are plots of the rise in recent years of artemisinin resistance. Artemisinin is one of the major frontline malaria drugs. And if parasites become resistant to these current antimalarials, then the world will have a problem. So you can see that actually this resistance has arisen in Africa in recent years as well. Right, so to get through this, let's get through these years. I'm going to divide my talk into three bits. I'm going to talk to you first a little bit about these classical variants. Then I'm going to talk about discoveries from genome-wide association studies, and I will talk about host-parasite genetic interactions. And if I have any time left, I will draw some conclusions at the end. So I can do this bit quickly because we've heard a bit about this already, right? The original evidence for genetic variants in susceptibility to malaria was this kind of epidemiological thing that some human traits seem to co-localize with areas where malaria was prevalent. So here's a couple of quotes on that from 1949 seems to be the date where this really kind of kicked in. So here it is, Montalenti recorded gene lethal in the homozygous state, so that means if you carry 2 copies of it, lethal, and it occurs in heterozygous state with such high frequencies that it must have been an advantage for individuals that carry it. So instantly, when talking about malaria, we're talking about selection, natural selection, the selective advantage. And here's a quote from Anthony Allison, who I'll show you data from in a moment, who similarly was talking about the same thing. So Montalenti observing frequencies of thalassemia, so that's blood disorders in malarious parts of Italy, and then he was working in Africa to study sickle cell disease. So here's data from Anthony Allison's study then, showing the rates of malaria parasitemia in sicklers and non-sicklers. So sicklers here actually means heterozygote carriers, sickle trait, as we might call them. And if you sort of work out the numbers here, you'll see that in this table of numbers, the malaria infection was about 2-fold lower in the AS individuals, in the sickle trait individuals, than it was in wild-type individuals. In this study, in this sample of about 290 children. Now, one of the things that we have to get at is that, that actually a study of 290 children like this, observational study, it's just not enough. And we've just heard a talk saying that really we should have a much larger number of samples for this kind of study. But as it turns out, this particular finding has been replicated many times over. So here is a more recent meta-analysis of work. This time it's not in infection, but in severe malaria. And so the upshot of all this is that really the effect size of sickle cell against severe malaria is something like 10-fold protection. So 10-fold protection is really important, right? So I guess everyone knows malaria. I've just sort of explained how important malaria is. Here's another sort of example of that from a study in Ghana, where in the least malarious parts of Accra, they estimated that about 1 in 20 children would die of malaria in the first 5 years of life. So if you have a variant that gives you 10-fold protection, that's 1 in 200 chance. That's really good news for you. All right, so this is the malaria hypothesis, malaria providing a positive selective force on the gene alleles. So sickle, obviously, if you have sickle cell disease, that's if you inherit 2 copies of the sickle cell, you have serious disorders as well. We've heard a bit about that. It happens like this. It's a mutation, a protein coding mutation in the hemoglobin beta chain. It causes hemoglobin to polymerize into these big long chains of molecules. They have the effect of turning your cells into banana shape, as we have to call it now. And you know, that leads to sort of lysing of the cells and then sort of into anemia and things like that. So it's a serious disease, and as we've heard, life chances are not very large if you're in a sort of rural setting in Africa. All right, so I just want to emphasize here that although that's kind of pretty well understood, that etiology, it's not really understood how sickle heterozygotes, how sickle trait is protected against malaria. So just to clarify that, there are several hypotheses for how that works, but it's not really clear which of those is primary or whether there are other things going on. That's a topic I'll come back to a bit later on in my talk. Okay, so malaria hypothesis, malaria pushing alleles to higher frequency and then something against it pushing it to lower frequency. We don't really have to speculate what would happen if this sort of balancing force of anemia were not there, because there's another classical malaria variant that's like that. We heard about it in Garrett's talk this morning briefly. Here's a map of the Duffy negativity allele, which provides protection against another form of malaria, Vivax malaria. And what you can see here is that essentially across all of Africa, the allele is essentially sort of close to 100% frequency. So this is one of the examples, primary examples of a selective sweep in human populations. The sort of molecular etiology of this is quite interesting as well. What's going on is that this gene, DARK, expresses a red cell surface receptor, which the Vivax malaria uses to enter red cells. But the mutation itself is not inside the gene, it's upstream of the gene in this piece of DNA that has a sort of GATA here. So GATA is a transcription factor that binds onto this particular DNA motif and upregulates genes in red cells. So the mutation involved here destroys this GATA motif, right? So it gets rid of the GATA motif, gets rid of the transcription factor, removes the transcriptional machinery, and now all these mRNA molecules that were coming off the gene no longer are, no receptor on the surface, protects against Vivax. Okay, so those kind of were my 2 classical examples, actually, that I should cover. And then I sort of ran into this problem that in the kind of, these discoveries led to a great deal of interest in finding new malaria genes, new genes that would affect his susceptibility to malaria, and indeed there were a lot of other alleles proposed. But coming from lots of different directions. So some studies of natural selection in the human genome would find genes that are under selection. Lab studies focused on particular genes. Remember, this is all before the human genome was published, so we're talking about candidate genes, essentially. Smaller epidemiological studies in hundreds of thousands. There have been a large number of them. I wasn't sure how to summarize them, but then I remembered that my former boss and, well, the late, great Dominic Kwiatkowski, Fellow of the Royal Society, had published this paper in 2005 that did my job for me in summarizing these. So here is a list of, in kind of 2005, this kind of list of genes that have been sort of implicated in malaria in one way or another. And it's a big, long list, right? So first of all, there's the blood cell ones up here that we've been talking about, HBS, other beta-globin mutations, the thalassemias, mutations of glycophorins, there's G6PD deficiency, which I haven't mentioned as another one, but then also kind of genes involved. Lots of people thought that cytoadhesion, so in other words, red cells sticking to other red cells or to the endothelium wall to try and keep malaria out of the spleen were involved, so a bunch of genes in that. And an obvious thing is immunity, right? Clearly, immunity is involved in malaria infection in some way, and so a large number of immune genes have been kind of proposed as being involved in protection. But the real picture from these is actually pretty unclear, and so Dominic sort of summed that up in his paper in this way. So here's the kind of sentence about that. When the same polymorphism has been tested in different geographical locations, the results have been variable, not simply failure to replicate, but in some cases the association would go in different ways in different study populations it was tested in. Right, so to summarize that, it clearly is a bit difficult. Now, Dominic's program at this time with his partners, with his global partners, was to say, well, let's sort this out. Let us start now a project that instead of doing sort of piecemeal pieces of work in different populations, we'll try and bring together a network of people to generate a large data set in which these questions could be answered across the whole. And so, and Dominic sort of became the spearhead of this thing that was called MalariaGen. So that's what I kind of want to talk about now. And its aim was to conduct a genome-wide association study in as large a sample as possible. So I'm going to just kind of skip ahead to the results. So MalariaGen is the name of the network that Dominic and the set of global partners from 15 countries worldwide put together. It did I'm skipping over a great deal of work in terms of setting up that network, organizing the ethics for it, capacity building, training, logistics of getting samples. Samples were genotyped in Oxford and in the Wellcome Sanger Institute, so they were shipped from Africa along with clinical data. There was a lot of interchange of people with training. Many students came to Oxford or Sanger to do the work. There were the issues we've heard about LD, so one of the pieces of work that the network did was to generate reference data sets. This number should say 800. So we added, in our study, we added these 800 whole genome sequences from population samples onto the available reference data, 1,000 genomes populations, because of this feature that Ambroise was talking about, the large amount of genetic diversity in Africa that was not represented in the reference panel. And so the culmination of all this then was a GWAS study which ended up including over 17,000 severe cases of controls from this set of populations, mostly from across Africa, and there were also some samples from Vietnam and Papua New Guinea. So that's I like to sort of call that a big sample size, but it isn't big, really. Really, we need bigger sample sizes, but it's the biggest one that we're going to get for a while, I think, in severe malaria. Okay, so I'm just going to go ahead and show you the results of that. Oh yeah, just to sort of mention, we've heard about sort of the high genetic diversity, and these are sort of principal components plots from each of those component populations, and sure enough, in the Gambia, there's a great deal of structure. It's sort of correlated with reported ethnicity. The same thing is true in Kenya. Cameroon has a great deal of structure in these data. All the populations essentially are highly structured in the way that we've heard. So these are the kind of this is the kind of overall result from this. So this is the type of plot that you'll be familiar with, is the so-called Manhattan plot, right? So we've got the human genome stretched out along the x-axis. Each point is a genetic variant, and on the y-axis we've got the evidence for association. So a crucial point to realize here is that the genome is very big, and there are lots of genetic variants. You need a lot of evidence to start to become convinced that one of these variants might be really truly associated here. So you need something like 10 to the 6 on this axis. And if you count these things up, you'll see that, well, we've got only a handful of things that are sort of meeting this strong criteria for for association here. So I'm going to kind of just go back to this slide for a moment and try and give you the answer to what was the answer to these things here. What I'll do is just shoot down the ones here that don't really have any evidence in this study. Looks like that. Right, most of these things, and in particular all of the things that have to do that were listed to do with cytoadhesion or immunity, bizarrely, just simply don't seem to have evidence in our study. Now, to avoid being misleading here, I'm not saying that immunity or cytoadhesion have nothing to do with malaria. They certainly do. But what this does say is that whatever's going on in those things, it's not simple enough that we can catch it in this kind of simplistic case-control type study like this. So there could be a lot of reasons for that. It could be environmental variation, it could be parasite variation, it could be difficult parts of the genome that our study just doesn't access. But it's certainly true that these aren't the biggest and strongest things sticking out there. What did come out of these GWAS studies, however, was new variants up at the top of the list. So I just want to show you a couple of those really quickly. So, two new variants essentially have been discovered by these GWAS studies. So one of them turned out to be not these deletions of glycophorin A and B, but duplications. I'll show you that in a moment. And then another one was this ATP2B4 calcium pump. So let me show you that one first. It's on chromosome 1. It's kind of interesting, right? So here's a picture of what ATP2B4 looks like. It encodes this calcium pump in the membrane of red cells. But actually it's expressed in all cells in your body in this form. So here's a picture of the gene. Its exons, those are the bits that get turned into mRNA, and its introns that get spliced out. However, in red cells, it turns out that it has a different transcript isoform expressed. It's got a different first exon, this thing that I've colored red here, and otherwise it's sort of pretty similar, but the different transcription start site. And that's important because just upstream of this transcription start site is, guess what, a GATA binding site, right? This GATA motif in the DNA. So what's going on here is GATA in red cell erythropoiesis, GATA transcription factor will be binding this GATA site unless this mutation occurs that has knocked out the GATA site, just like for the Duffy negativity allele. Okay, so it goes this way. The risk allele has the GATA site, so expresses the gene. The protective allele, malaria protective allele, disrupts the GATA site and does not express the gene. Now that's a bit of a puzzle as well. So here's a kind of schematic of how you might think of ATP2B4. It's this calcium channel. It sort of helps with calcium homeostasis, and it's moving calcium ions out of the cell. And so it would look a bit like this. The risk genotype should have lots of those, probably less calcium. The protective one would have fewer of these, probably more calcium inside. This is weird, though, because parasites like calcium. Parasites use calcium a great deal in their sort of growth and signaling and growth as they go through. But there is a sort of solution to this, and incidentally, this isn't something that's been tested, I don't think, but it's a sort of speculation, which is, of course, that as that parasite goes into the red cell, it pulls a bit of the membrane with it, and it ends up in a vacuole inside the red cell. And of course, as it does that, it inverts the red cell going in. So probably what's going on, I think, this is my hunch, is that this vacuole ends up with these calcium channels, but now in an inverted form. So this then sort of explains this version of this direction of effect that we see for this thing. But as I say, this hasn't been tested, but I think it's a good hypothesis. Okay, so that's ATP2B4, this red cell-specific expression, somewhat like the Duffy allele. So I just wanted to quickly talk about this second thing. These are these glycophorin duplications. I'll do it very quickly, I think. Glycophorins are, well, there's 3 glycophorin genes arranged tandemly on chromosome 4. Most people in the room, most chromosomes in the room, I think, will look rather like this. There'll be 3 glycophorin genes on those chromosomes. But it turns out that in parts of East Africa particularly, people sometimes have a different structure of that part of the chromosome. They have this structure. So not three genes, but 5 genes. And actually, not just 5 genes, but these genes are sort of hybrids. So that's important because these genes encode transmembrane proteins in the red cell, things that stick out of the red cell and they have tails sticking into the red cell. This thing that encodes what's known as the DAN2NE blood group produces something like this. It has an intact copy of glycophorin A, which is this very common molecule on the surface of the red cell, but it also has these kind of hybrid glycophorin BE things. So we just heard a talk about these, about variants being different in different populations. I should just emphasize that this is an extremely East Africa specific variant. And actually the only reason that we were able to access that in our study was because, let me just travel back in time a little bit. Was because back in our sequence data that had been collected for this study, we happened to have samples from Tanzania in the east of Africa. So that's over here, this kind of orange dot over here. And among those Tanzanians, 8 of them happened to carry this particular genetic variant. But if you look anywhere else in the world, including in all the other 3,600 other individuals in this, in the reference panel, we don't see that genetic variant, right? So we had to get the right populations in order to see this thing at all. Okay, sorry, I'm just going to zoom forward again 3 times. That's pretty interesting. We were really excited by this because, of course, these things stick out of the red cell, and it's known that falciparum parasites bind onto these things during invasion. There are sort of specific parasite genes which are known to bind onto them. We were sure it must have been about that. But a team in Kenya then went to do a functional study of it, and they concluded something different. So this is an amazing paper, actually. It's a really good read. They were able to, for example, watch in real time, watch parasites invading red cells and count the number of invasions into the Dan2 cells and into the non-Dan2 cells, along with a number of other innovations in how these were measured. What they actually discovered, think is the case, is that this all has to do with membrane tension. So somehow these Dan2 things have probably more of the kind of intracellular tail. They have stiffer red cell membranes. And guess what? If a parasite is trying to get into a bag, it's harder to do it if the membrane is stiffer than it isn't. So possibly nothing to do with this sort of specific interaction with receptors and ligands at all, possibly to do with the stiffness of the thing. I think I could have guessed that in the start, actually, but I was slightly underwhelmed. But it was interesting. Okay. So by doing this kind of study at large scale across multiple samples, it provides a way of resolving these type of questions. Were these signals real or weren't they real? Are they strong enough? How strong are they relative to other things? Which ones should we get excited about? Are there any new things? And so this plot just is sort of illustrating that. So you can see the strong effect of sickle hemoglobin, this sort of tenfold protective effect. The ones we were just talking about, the Dan2 variant, are sort of the next strongest. They have something like a 50% protective effect if you have 1 copy, and probably more than that if you have 2 copies. But only seen in East African populations. And then these kind of other variants around are kind of tailing off in terms of how strong their effect is. But at the other time, you have to recognize that the frequencies vary a lot as well. So, for example, the O blood group has a modest level of protection, but 50% of Africans have O blood group. So that's important. Okay, right, just to mention that heritability was mentioned last time. It is true to say that the estimates of how heritable this phenotype of severe malaria is come out at about 20%, both from our genome-wide association study and from other family studies. The variants that we identify so far explain something like 2%, so that means there's stuff left to discover in the genome. But I want to stop on that point just there, because I want to move on to talking about host-parasite genetic interactions. Because obviously, everything I've been talking about so far has missed the elephant in the room, really. So we know that these human genetic variants are protective. There's a sort of subset of them here with their effect sizes. We've known some of that for a long time. But of course, the malaria parasite is also has a big genome. It's a 23 megabase genome. It's 150th of the size of the enormous human genome. It has lots of genes in it, something like 5,000 genes, and there are millions of genetic variants. And we know that parasites evolve quickly. They evolve over timescales much quicker than humans because their life cycle is on the order of months instead of tens of years. And they have adapted in the past, for example, to quickly to pressure from drugs. Right? So an obvious question here is, well, if these human variants are around in the population, protecting against malaria, preventing parasites from infecting, invading red cells, replicating or transmitting onwards, they must be placing selection pressure onto parasite populations, just as malaria was placing selection pressure on human populations. So have the parasite population adapted? What would it look like if they had? So that was the sort of starting point for our host-parasite interaction study. Now, I've got to do this quickly because I'm going to run out of time. I just wanted to show you kind of how we attacked that. We've done a particular type of analysis. Others, you could think of others. We have our GWAS study that I've just described. We know the human genotypes for a whole bunch of people, children who were in hospital with severe malaria. And so if you take a blood sample from someone with severe malaria, they have human DNA in it, but they also have malaria parasites in their blood. So they have parasite DNA in that blood as well. So that means you can sequence the parasite genome from those infections there. So that was the idea. We would sequence those parasite genomes, called parasite genetic variation, and then we were going to look for association between human and parasite genetic variants. So let me just show you quickly how that went. We took samples from our GWAS study, but we took the ones from the Gambia on the west of Africa and from Kenya on the east of Africa. We went to a process either just directly sequencing all the DNA for the high parasitemia samples or an amplification process, selective whole genome amplification for lower parasitemia samples. And what I'm just showing you here is just trying to convince you that it produces usable, at least, maybe good data, right? So what's plotted here is a region of the parasite genome, and the gray bars here are sequence read coverage. So how many genome sequence reads did we get back across those things? So if you're used to looking at human data, you'll probably think this looks a bit rubbish. It's very variable. It goes up and down a lot. That's actually due to the kind of high AT content of the parasite genome. It's harder to sequence. And the amplification of things don't work perfectly. But what you can see is that across a range of measured parasitemia levels, including some that were reported as not having any parasites in, we recover parasite sequence reads, and it's has a kind of high coverage, at least across some genes in the genome. Okay, so good data, right? So we use this for sort of variant calling, quality control, genetic variant calling, and then overlapped it with our human samples. So discovery analysis of just over 3,000 samples. And then we did the simplest thing that we could with it, which is to test for association between the parasite genotype, so pairwise between parasite genetic variants and human genetic variants, putting the parasite genotype as the outcome variable, the human genotype as the predictor variable, and we only have one covariate in this particular scan, which was the country. Remember, there's Gambia and Kenya, and that's because we didn't see much structure, actually, in the parasite populations within countries. It didn't seem to need it. Just to clarify that we focused for this study on a number of candidate human mutations, so all the ones that I've described earlier, a longer list of candidates from the GWAS, other blood group variants and other blood group genes, and we also put HLA immunity alleles in there as well. In the parasite genome, we focused on all of it, but there are bits of the parasite genome that are difficult to get at with short read sequencing, so those bits are sort of excluded. Telomeres, subtelomeres, highly repetitive bits. So that just sort of makes clear what we've got there. Here's what the results look like. So it's another Manhattan plot now for the parasite genome. The parasite genome, 14 nuclear chromosomes stretched out here. Each point is a parasite genetic variant. The y-axis then is evidence for association with one of our human genetic variants, averaged over them. And you can instantly see that we have signal right here. So 3 regions of the parasite genome have strong evidence for being associated in these severe cases with human genetic variation among the set that we discovered. So here's what it looks like then in the human part of the study. Weirdly enough, across all these human variants that we included in our scan, not 3, but just 1 human variant turned out to be associated with parasite genetic variation. And I wonder if anyone can guess what the human genetic variant that stuck out here was. I think someone said it was sickle hemoglobin. That strongest effect, right, the thing that was strongest protection against malaria, lo and behold, turns out to be involved in this interaction. Okay, so we've taken to call these this PFSA, Plasmodium falciparum sickle-associated loci 1, 2, and 3. And just to kind of show you what those regions of the parasite genome look like, they look like this. So the pluses here indicate that actually the strongest signals here are protein coding change mutations, the SNPs in there. in all three of these regions. Down here, we sort of plotted the genes. This one has a name, acyl-CoA synthetase family member 8. There are something like 13 acyl-CoA synthetase family members. Nobody quite knows what this one does. These two genes actually don't have names, really. They just sort of have numbers. This is a putative exported protein, means gets exported from the parasite into the red cell. And this thing's a putative tyrosine phosphatase, but these are all like in silico predictions of them, right? So we're instantly in this problem that we don't really know what these genes do. And so I'm just going to show you, that's a table of counts. Let me just skip to this version for simplicity. So if you remember that, that idea that sickle provided a 10-fold protective effect that I described at the start. But look what it looks like now. So if we divide up that 10-fold protection across the different genotypes of the parasite at the three loci, the 2 on chromosome 2 and the 1 on chromosome 11, here's reference-like parasites. Our study estimates that there's a very, very strong protective effect, 1 in 100 protective effect, more or less, against these sort of reference-like parasites. But maybe there is a much weaker effect against parasites that carry these sickle-associated alleles at all three of these loci. And perhaps if the parasites carry kind of a mixture of these things, then maybe the effect is somewhere in between. Now, this comes with a lot of caveats, right? We're only doing it in one study. We'd like more data to really make these estimates. This is the same study that we discovered the effect in. But nevertheless, it shows you that there's some strong interaction going on between sickle on one hand and these three loci on the other. Okay, so let me just kind of finish that off with a little bit of population genetics, if I may. So I'm hoping I've convinced you that this story that started in 1949 and the discovery of sickle protection has new features when you start studying sickle against parasite genomes. These loci turn out to be interesting for all the same kind of reasons that the malaria-associated human ones were, namely natural selection. So here's a plot of frequencies across populations roughly ordered from West Africa to East Africa and then non-African populations. And you can see that these three variants in three parts of the genome are, well, they're not really there outside Africa, and they vary a lot within Africa as well. And they sort of co-vary within Africa as well. But the one in the middle also isn't really seen for some reason in West Africa, despite being at high frequency in the Democratic Republic of Congo. Okay, this is data from MalariaGen PF6, so they're community sampled infections. If I replace this x-axis with an estimate of the sickle allele frequency in humans in those populations, you can see that, sure enough, we get kind of a correspondence here. So this is a sort of population-level replication of this effect that we'd seen inside severe cases. Okay, so I've got no minutes left. I want to just show you one other really odd population genetic feature of these things, and then I will let you go to lunch. Here's the really odd thing. Malaria parasites, just like humans, undergo meiosis. This happens during their passage through the mosquitoes. So meiosis means independent segregation of chromosomes, and it means recombination. It breaks down linkage disequilibrium. But what we see for these loci is that despite this feature, the variants, despite being on two different chromosomes, chromosome 2 and 11, are highly correlated to each other. So you can see that here in our Kenyan severe cases, 80% correlated, a bit lower in Gambia. If you look instead of the severe cases in community sample infections, we see the same pattern. Here's this MalariaGen PF6 data. So for example, here's parasites from Ghana. There are 807 in the sample. The chromosome 2 and chromosome 11 loci are 86% correlated. And it's the same pattern across all these African populations, although for some reason that we don't understand, Gambia has a lower level of correlation, but still there. So this is extremely unusual because of this feature of recombination, right? So here's a kind of, just to kind of emphasize this, here's a histogram of all pairwise LD values computed between variants on two different chromosomes in the parasite. And it looks like it ought to look. All the variant values are close to the kind of zero line, but there are a few pairs of variants that escape, pairs on two different chromosomes that escape this. And it's really these sickle-associated variants that escape this the most, and they escape it in all these different populations. And just to note that, you know, the next ones down the list, or in Gambia, the top of the list, turn out to be well-known drug resistance mutations. So these things that we know are under strong positive selection. So this is I've added a question mark here because this is just a big puzzle. I mean, I think it's safe to say that none of us understand how it can be. Perhaps there's something like epistasis going on, but we also got to be conscious that there's population structure. All right, I need to wrap up. So I think of this as a bit of a reverse malaria hypothesis now. So instead of malaria prevalence pushing selection for human alleles, we've got human alleles pushing selection for malaria alleles instead. And so the hypothesis here is that the presence of sickle in the population positively selects for these particular malaria alleles, and that must be one reason that they are spreading. That doesn't explain why they haven't fixated in the population yet. They are at intermediate frequencies, and we don't know what other forces are involved. And you know, it's also raises a whole bunch of other questions. We don't know what the biology of these genes is or what the variants do to them. We don't understand this LD signal. But there's a hope that by kind of using these parasites, we may start to get more answers to how does sickle provide a protection. Right, now I promised to go, so that's taken us to 2021, and I promised to go to 2023. There is an extra bit to this story, which I'm going to tell you the story, really. It's a very recently released paper on bioRxiv, hasn't been peer-reviewed yet, in Ghana. So this is in mild malaria cases in Ghana. It's a paper by Lucas Amange-Etedo and from Will Hamilton in Sanger. They discovered what seems to be a fourth locus associated to sickle. This one is a bit the opposite of that middle variant because it's only seen in West African populations, not in East African populations. So there's something interesting going on with this as well. Okay, right, so this is what we're hoping for, you know, in future as we go off to the right of this slide. I haven't mentioned mosquitoes at all, just to say that this is a missing part of my talk. There is a lot of work going on mosquito populations, and it's equally kind of interesting and fascinating, but I'm not going to say anything about it. So I just wanted to end by just thanking all the people that have been involved, the performance has gone here, and in particular, Dominic obviously has played a giant role in all of this, and sadly passed away earlier this year, which is a shock to many of us. But I want to thank all the people that were involved in the study and all the people that contributed, and thank you for listening.